Good morning, everyone. Welcome to the second day of the Samuel M. Nabrit Conference for Early Career Scholars. It's great to see everybody this morning. Thanks for making an early start, especially for people who had to travel this morning. We do have a little bit of excitement in the schedule due to some travel difficulties, and so our panel on institutional culture change this morning will feature 2 in-person panelists and 2 remote panelists who will be joining us from the lovely city of Charlotte, North Carolina. And we really appreciate their fortitude in joining us even while they are stranded away from home and not here in person with us. Mark Johnson will actually introduce that panel. We understand that the panelists have the Zoom link and are in good shape to join us for the panel event. Mark will also introduce and give some information about the poster session, which he's been organizing, so he'll do that right before the panel event. So without further ado, I am delighted to introduce the first speaker of the morning, who's Karina Vargas, coming from the Marine Biological Laboratory in Woods Hole. And she is going to talk to us today about alpha-synuclein participates in clathrin-mediated SVE through regulation of the AP2 complex. And let's go out here. And you are right there.
Thank you, Alison.
And you got the pointer, right? Good.
Good morning, everybody. Thank you for having me today. So today I want to talk to you about how synuclein participates in clathrin-mediated synaptic vesicle endocytosis. But first, I want to tell you why synuclein, synaptic vesicle endocytosis, and that these 2 processes are very important for Parkinson's disease. So here I show you a very basic cartoon where, I don't know if you recognize, these are synaptic vesicles that are filled up with neurotransmitter. And when a nerve impulse comes to the end of a neuron, these synaptic vesicles are fused to the membrane to release the neurotransmitter and continue the communication with the next neuron. So this process is called exocytosis, and when all these membranes that are now fused here, they need to be recaptured to produce new synaptic vesicles and continue different rounds of neurotransmission. So I don't have to tell you that this is very important for everything we do in life. I'm very interested in the internalization of these synaptic vesicles. It's a very complex process that requires different proteins, and especially this protein called clathrin that produce this sort of cage around the synaptic vesicle that give the shape and the size. And then there is an extra step here where the neck is cut. So now the clathrin-coated vesicles is internalized. There is an uncaging that produces now the synaptic vesicle that is refilled. And why this is important for Parkinson? Because in Parkinson, around 10% of the patients have this form that is inherited. It's early onset, and it has mutation in different proteins. And a very big subset of these proteins are involved in the uncaging of the synaptic vesicles. As example here, oxalin, LARC, and synaptogenic. But then also the protein that I am interested in, synuclein, is important for the initial steps of this process. So I work a lot on putting synuclein in this part of the cartoon, and this started 10 years ago when there was a lot of controversy about what it was the physiological function of synuclein. So there were many different mice knockouts that have single mutations, and the data was controversial. So I decided to use the triple knockout, meaning that in the family of synuclein, there are three proteins, alpha, beta, and gamma, and they're very similar, as you can see here, in the alignment of the sequence. So if we observe the first part of the protein, is very, very similar, and this is a very important part of the protein because it folds into alpha helix, and it binds to lipids, just synaptic vesicles, and also flat membranes, and that's very important for the function of the protein. So the first step I did, it was purifying these wild-type neurons and triple knockout, and I transfect them with this very special protein called synaptofluorine, which allow me to monitor exocytosis and endocytosis. And how it does it is because it's a GFP that is attached to the transmembrane part of big glut that allow it to be facing the lumen of the synaptic vesicle where the pH is acidic. So in those conditions, this protein is quenched, and when you fuse the synaptic vesicle to the membrane, now this protein is going to be in contact with the extracellular media. In the extracellular media is around 7.4, so it will increase the fluorescence of this protein, and we can monitor exocytosis. The opposite happened too when the synaptic vesicle is internalized. Now the lumen of the vesicle is going to be reacidified, and there is going to be a quenching process, and we can monitor endocytosis as a decrease in the fluorescence. So I did that, and I compared wild-type neurons with triple knockout neurons to see the effect in the recycling. And I found that the endocytosis after stimulation, or this decay that you see in blue for wild-type, if we compare it with the triple knockout, now the triple knockout had a delay in this internalization. And through this experiment, but many others in this paper, I showed that synuclein regulates synaptic vesicle endocytosis. Now going further to see what is the molecular mechanism, because I'm that kind of scientist, I'm looking for these sort of things. Synaptic vesicle endocytosis is mediated by clathrin, as I told you before. And one way to observe how this happened is see how the polymerization on the membrane happened so this process goes forward. So I asked myself, is synuclein controlling this process? And I did a very simple biochemical experiment where in a little well that has a cytosol in the well, I grew a monolayer of lipids, and then I allowed this cytosol that contain all the proteins from the brain to polymerize clathrin on this membrane. I observed this by electron microscopy, and you can see here how this beautiful sort of honeycomb is the clathrin formation on the membrane, and we can measure the size. So then, instead to use just wild-type cytosol, I also compare what happened if we have wild-type cytosol and we measure certain size, and then we have the triple knockout cytosol that doesn't have synucleins, and we see that now the size of these patches are smaller. So synuclein would be controlling the polymerization of clathrin on membranes. And I partially rescue introducing to this system mouse alpha-synuclein. So clathrin, as I told you, it polymerizes on the membrane, but it also needs a special proteins that we call adaptors, because they adapt clathrin to the membrane. Clathrin doesn't have lipid binding domains, so it needs these adapters, which is AP2 and AP180. So I wonder what happened with this protein. Is synuclein affecting these proteins, or is it affecting clathrin itself? So what I knew from my previous paper is that the adapter AP180, it immunoprecipitate together with AP180 and SV2, so it formed part of a complex with this protein. And it also, recently, I found that the AP2 complex, which is a heterotetramer, so it's a more complicated protein, it also interacts with synuclein, and I can show you here that it pulled down with just this part of the protein, the beta appendage and the hinge, and it also partially pulled down with this part of the protein, which is the core of the protein. And also I did my control, as I showed you before, for AP180. So these two adapters that are very important for the polymerization of clathrin interact. I don't know still if directly or indirectly with synuclein. But now what they do is bind to the membrane. So I wonder if synuclein regulates somehow this binding of the adapters to the membrane. And to answer this question, I purify synaptic membranes from synaptosomes from just a wild-type mouse. And then on the other side, I purify all the proteins from the brain, the cytosolic proteins, which we can say they are all of them. And then I mix them up to see how many of these proteins now are binding to these synaptic membranes that I also clean up so they don't have any other protein binding to it. And then my readout of the experiment is how much of this protein now are binding to these membranes. My experiment is to take synuclein from this mix of proteins, and then we have a control versus a mix of proteins that have all the proteins of the brain but synuclein, and see what happens. So as you can see here, with my treatment of the cytosol, AP2 and AP180 remain in the same levels. These are the membranes that I clean from any binding of other proteins. And when I mix the membranes with the different cytosols, you can see here in the control that AP2 binds to the membranes, the synaptic membranes, AP180 does too. But when we don't have synuclein in this mix, now AP180 is still there, but AP2 is not properly attached now to the membrane as in control conditions. So here I just show you the quantification where we can see the significant decrease in the binding when we don't have synuclein in this mix of proteins. So my conclusion from these three different studies is that synuclein somehow regulate the binding of one of the adapters, even if it's in a complex with both of them, to regulate clathrin, and in that way, it could be regulating clathrin-mediated synaptic vesicle endocytosis in neurons. So this is sort of like my hypothesis, and there is more work to do to figure out if this is true. And but my first next question, like sort of the small question, is like if these interactions are direct with AP2 or indirect through different other proteins, and their regulation could be like synuclein could be affecting other proteins before that affect the recruitment of AP2 to the synapses. And with that, I want to say thank you to my lab at the MBL, especially to my mentor, Jennifer Morgan, to my funding, and also some of this work was made at Yale several years ago. And I want to also extend the invitation for everybody to go to the MBL because we have amazing programs in the summer for every stage of your career, for undergrad, grad student, postdoc, and also for faculties that want to make research in the summers and use some of the marine animals. So I don't want to leave without saying what I do with a marine animal because the work that I've shown you is just a mouse. But I use this animal called the lamprey, the sea lamprey, and it's very spectacular because it has very big synapses. And it gave me some advantage compared to the synapses of the mouse that I used in the past, because in these synapses, we are able to see nanodomains in the presynaptic compartment. And I show you here a graph where the active zone, where exocytosis happen, it could be visible and differentiated from the periactive zone, which is where endocytosis happen. And here I show you just a simple confocal. This is not super resolution, where we can see the endocytic zone, that is this ring of actin that I can mark around the synaptic vesicles in red. And on the other side also, we can see in the cluster of synaptic vesicles already nanodomains of synuclein. And this is, for me, is an amazing tool that I am planning to use to characterize these endocytic nanodomains, and also to evaluate how this sort of like mechanism that I'm describing in these mouse systems, how synuclein, AP2, and the lipids interactions are important to maintain these endocytic domains and maintain also the functionality of the synapses, which is very important for prevent neurodegeneration, as I am studying Parkinson's disease. And thank you.
Excellent talk. So I was just wondering about the actin ring. Is that something that's shared with other types of endocytic vesicles, or is that unique to the ones that you studied?
Recently, a researcher that, I don't know if you know about the ultra-fast endocytosis, Shiyaki, which was the guy that discovered this, he also have done some experiments and show in mouse synapses this ring of actin, which for him is a challenge. It took so many years because of the size of the synapses. So he is like doing this work and it aligned with ours, but still I think like the lamprey synapses is way more easier. Like you don't have to make as much of an effort that he is doing now to see these like very simple compartments, but it's good that he also saw it.
That was a very nice talk, some beautiful images. So every neurodegenerative disease has its own topography. Certain diseases start in certain neuronal populations. And with Parkinson's, the pathology starts in the substantia nigra dopaminergic cells. So is there something different about that, the presynaptic vesicle recycling in that cell population that makes it more vulnerable to synuclein?
Yeah, so the dopaminergic neurons, they are pacemakers, so they are constantly triggering releasing neurotransmitter. So that in the field, we think that make them more prone to suffer neurodegeneration. But then there are other studies nowadays that also are studying other kind of neurons that could be affected a little earlier than the dopaminergic neurons. Because there are other, like when we see what happened to the patients, when the patients have 60 or 70 years old, they are having this motor disorder part of the disease, which is related to the dopaminergic neurons. But with the studies of what happened to this patient earlier on, they have a lot of disturbances in sleeping, depression, constipation, things that we think that could be related to the anomalies in the presynaptic compartment of other neurons. For example, the neurons that are from the stomach to the brain. And that's very important to sort of pay attention to it, even if these neurons are very, very sensitive. Like the dopaminergic neurons are very sensitive to Parkinson's disease or anything that could cause it. But yeah, that's the reason, I think.
Okay, we have time for one more question before we move on.
Yeah.
Hi. Yeah, working. Very nice talk. I'm curious about the three forms of synuclein. And in the lamprey, is there a single form? And does that help you to figure out maybe what the overlapping roles for the three forms in the mouse might be?
Yeah, so the lamprey is always used like as an ancient animal in evolution, because many of the proteins that we have 3 versions or 4 versions, the lamprey always have one. And in this case, lamprey express gamma-synuclein, which is the letter for the last one, but is more similar to alpha-synuclein. And we think that there was a duplication and triplication of this gamma-synuclein to produce these 3 members of the family. But on the other side, lampreys never get Parkinson's, so it's not like it's like alpha-synuclein itself, like perfectly. But we can recreate Parkinson's disease by acutely injecting human alpha-synuclein in the synapses. We observe disturbances in cluster-mediated endocytosis and neurodegeneration, which that's another perk of the lamprey synapses. Like when we use mouse, the modifications that we do in the knockouts cause a lot of compensations, and that's the reason I'm sort of walking away from the triple knockout mice. That is, for me now, it's a tool, but it's not the main thing that I want to study, because now when you take the spinal cord, you can inject elevated amounts or mutants or different drugs and just wait 10 minutes and then see what happens. So you see the acute changes in the synapses right there, and you don't have to wait, you don't have to see how other proteins overexpress or decrease. That sort of like make your readout of the experiments not clean. Thank you.
I wasn't sure.
Come on up. So our next speaker is Nova Pichesha from Harvard University, and she will talk to us about engineering the modularity of an alpaca-derived single-domain antibody fragment that targets class II MHC to induce antigen-specific tolerance.
Thank you. Sorry, it's really mouthful title. Cool. Hi. Well, good morning, everyone. First of all, I would like to thank everyone here for the opportunity to share my works, as well as, listening to me rambling for the next 15 minutes about my work. So I worked on developing strategies to induce antigen-specific tolerance, in particular in the context of autoimmune diseases. So a little bit about immune diseases. As you all know, in the midst of the pandemic, like where we are right now, our immune system is really important in fighting against pathogens, protecting us from the outside world. However, in about 10% or more of adult populations, in particular among women, there are our immune systems could be attacking our own cells, our own organs, our own body, leading to a manifestations of autoimmune diseases. As you can see in the list that in this little cartoon here, our immune system can virtually attack any organs in our body. So unfortunately, though, the current therapies for autoimmune diseases are kind of inadequate. So what we have been prescribed are immunosuppressive drugs or biologics that suppress the entirety of our immune system, leaving patients exposed to pathogenic attack. So what I would like to develop is a strategy in which we would like to just delete the unwanted specific immune response to cure autoimmunity. So a phenomenon we call antigen-specific tolerance. An example here is multiple sclerosis, in which it's an inflammation in the central nervous system. So if you look closely at the cellular level, we have immune cells that we call it other immune cells that we need to fight pathogens, while at the same time, there are also myelin-specific immune cells. So immune cells that can attack your own cells, creating havoc in the central nervous system, and you then see the clinical manifestations of multiple sclerosis. Actually, as a PhD student, I have developed, is this better if I stand like this? Because it's kind of awkward. Can you guys all hear me? Good. I think I'm much more comfortable speaking like this. I am pretty loud, as you can hear. So yeah, as a graduate student, I developed an engineered red blood cell method to deliver antigens and induce antigen-specific tolerance to protect against autoimmune disease. So it was great. However, a little bit about me. I'm originally from Indonesia. It's a country of 16,000 plus islands and not necessarily great in biotechnology. So if you think about it, a treatment of autoimmune diseases using cells will be a bit tricky to actually employ there. It's a beautiful country though, so if any of you guys are interested to ever visit, I have a great list of places to go and definitely can help you travel there. So yeah, so when I graduated from my PhD, I would like to still develop a strategy to treat autoimmune diseases in an antigen-specific manner. However, with some additional characteristic, for example, for the treatment to be cheaper, to be easy to scale up, to be easy to distribute. Hopefully a single dose can give a long-term protections, and the compositions can be modular enough that we can address multiple diseases and can work across immune genetic background MHC allotypes. So I don't use red blood cell anymore. I will introduce two different technologies that I've been using in this strategy. The first part of the technology is nanobody, or VHH. It's small but mighty, because if you think about traditional antibody that is out there, you can take a look into this. Here you go. Yeah, the recognition site that bind to specific protein comprises of two different domains, like the orange and the yellow. However, this is the kind of antibodies that us, human being, as well as mice have. However, camelid families, such as llama, alpaca, as well as sharks, interestingly, also have this heavy chain-only antibody. What that means is that the recognition site to a protein, the binding site to a protein only comprises of this one domain. So we can derivatize it further to make this VHH or nanobody. It's much smaller, so it's more stable and also more soluble. So it's quite an attractive delivery for our purpose. And I know if it is small, usually people said that it might be a disadvantage because you have a short circulatory half-life. However, in this particular applications, what we would like to do is going into the immune system, rewire them, and get out as soon as possible to reduce possibility of toxicity or unwanted subsequent side effect. And lastly, but really, really importantly, because it's much smaller and doesn't have any post-transient modification and so on, this is much easier to produce, and you can produce it in E. coli or bacteria, so it's very cheap. The process, just quickly to go through it, is that I'm making a nanobody against class 2 MHC product, so I immunize an alpaca. No worry, no alpacas were harmed, except for a little bit of injection site that they usually are not happy with in this process. And then we can make a library, a fetch library containing the nanobody sequences that we would like to then screen against specific protein, and we got the nanobody being expressed finally in a bacterial system. So this is just to show you the nanobody that we created is indeed specific for MHC class II protein. So why MHC class II protein is important here in the context of immune system? Because all of the professional antigen presenting cells, so the immune master regulator to like tell the adaptive immune system what to do, actually express MHC class II protein. So this just to show you as well a flow cytometry assay that what we are creating against MHC class II are specific for this protein. Then the other side of the technology that we are working and combining to this nanobody system is sortase mediated bioconjugation of nanobodies. So sortase is a bacterial transpeptidase that can recognize a specific amino acid sequence. So in this context is an LPXTG motif. So all of the nanobody that I've created has been engineered to carry this motif. And when we mix this nanobody with sortase, sortase will cut between the T and the G. And then if we provided the system in a mixture with the GGG carrying moieties, it can be peptide, it can be drugs, it can be fluorophore, or anything that you can think of that you can engineer to carry GGG, the resulting outcome of the reactions is the nanobody with the moiety or the payload covalently connected to each other with an LPX-TGG bridge. So that's the technology that we use. What we use it for is to make this, I hope you guys can see it, to make this adducts. So the adducts that we have is an adduct that comprises of a nanobody against MHC class II protein with the disease-specific autoantigen or self-peptides. So for example, in the case of autoimmune, in the case of multiple sclerosis, it's a myelin-derived peptide. In the case of type 1 diabetes, will be a pancreas-derived peptide, and so on, as well as anti-inflammatory drugs such as glucocorticoid. So these are covalent touch each other. So what will happen is that when we inject this nanobody peptide drug adduct into the circulation of a mouse in the preclinical mouse models, they will find an MHC class II protein on the surface of antigen-presenting cells. And what happened then, they will be, because MHC class II protein is being recycled very regularly inside these specific cells, they will be also taken into endosomal compartment, being digested by proteases. So this peptide will be detached from the nanobody, and it will actually be processed such a way it can be presented in the proper context MHC class II. So as you can see, it will go here carrying the peptide of interest. And because we also deliver glucocorticoids, the glucocorticoids will rewire the signaling within these cells such that the cells instead will start exhibiting more inhibitory signal as well as secreting anti-inflammatory cytokines. And the further outcome of it is that because of these engagements, then when autoreactive T cells that can recognize this specific MHC class II peptide context, they will, instead of their proliferating and being activated, they will then, some of them become regulatory T cell, some of them actually being deleted and being exhausted, and then that's a stage of antigen-specific tolerance. So just to show the data, we actually use this one. First, we use the multiple sclerosis preclinical mouse model. We can induce multiple sclerosis-like symptoms in wild-type mice by injecting or immunizing these mice with myelin oligodendrocyte glycoprotein peptides with a strong adjuvant to activate the T cells that recognize this peptide, as well as pertussis toxin to break the blood-brain barrier such that this T cell can go to the central nervous system and create inflammation there. Within 10 to 14 days, you will start seeing these symptoms. So, and then we can score them clinically from limb tail and greater degree of paralysis developing over time. So we can score from 1 to 5. So this is the result. What we have is that, so for example, if we inject our adduct 7 days prior to induction of the disease, so think of this as a prophylactic treatment, you see in red, so this is the proper adduct, comprises of the nanobody and the myelin-derived peptide. You see that the average clinical score is 0. So all of these mice are completely protected from the disease. And I can tell you that this is a durable protection because this adduct, the nanobody adduct, only lasts in the circulation for an hour or 2, whereas we induce the disease 7 days later. And of course, as a control, we have delivered the right nanobody but with the wrong peptide, or the wrong nanobody with the right peptide, and you see that they are all developing disease to completion. To prove further that we can do like a very long-term protections, we let these mice that you see here to keep going, and then once again immunize them with the protein, the mock protein, as well as the pertussis toxin and the adjuvant. And you can see that you have to at least some point stop the experiments because you want to summarize the paper and then get out, So we finished it at day 100, and then you can see here that they are still protected from disease. However, like everyone else always said, in the context of autoimmune diseases, we only treat patients when we see symptoms. So here is a therapeutic efficacy that I would like to show you guys, but maybe we can skip it through just this side of the graph. So let's say you wait until the mice reach level 3 of clinical score, meaning that if you see the mice in the cage, they have complete hind limb paralysis. So they actually have to crawl inside the cage. So if you just treat with one dose of the adducts, the mice will recede to like lower clinical score. So actually within 24, 48 hours, it's amazing how they can run around in the cage and being really happy. And gain weight too, and muscle. So that's actually something that we are really excited about. And like I promise you, I want to develop some things that can be modular, meaning that we can apply to different diseases accordingly. So in the left is a type 1 diabetes mouse model. So we could apply this to type 1 diabetes by changing the peptide of interest to a type 1 diabetes associated peptide. And you can see that all of the mice that treated properly develop normal glycemia throughout the observed timeline. And the context rheumatoid arthritis changed it to the rheumatoid arthritis associated peptide, and you can see here that they also have less joint inflammation. You can see it from the size of their paw. So I think at least in the preclinical mouse model that we have shown, we can develop some things that cheap, easy to scale up, distribute. A single dose can provide some kind of long-term protection and can address multiple diseases with slight modulation, of course, across all of MHC allotypes, immunogenetic background in mice, because all of the mice that mouse model I've showed you, they actually have different immune background. So, hooray. So I guess, of course, there are still much to do. Actually, when I submit the paper, some of the reviewers said, this is a lot of data, but there are much more that you can do. So of course, I agree with this person. So this is a conclusion that we could apply this for a lot of different diseases, and we are really excited about it. But there are a lot of other applications that we can envision this work can be applied to, let's say in the rejection against organ transplants, or in allergy or other chronic inflammation. But the mechanism, even though we have looked into it a little bit, there are much that can be done to like really fine-tuning it, and we are also investigating the safety profile of the approach and hopefully translate it to non-human primate and human. And in order to do that, partially, we also recently I co-founded the company based on this technology called Cerberus Therapeutics. We are hiring in case anyone here are looking for a job. I'm sorry, I'm really shameless on that. So thank you for everyone for listening, and also for my PhD advisors, or my postdoc advisor, and everyone who have helped me to complete this project. And I have like 2 minutes to answer questions. Thank you.
Great. We have time for a couple of questions.
Thank you, Dr. Pisesha, for an amazing talk, and I really like it.
Thank you.
I have 2 short questions. The first question is about phage displays. So which library did you use, and did your lab create your own library?
Yes. Yeah. So I immunized an alpaca with the protein of interest, and then we created a library from whose sequence we derived from the peripheral blood mononuclear cells that we got from the alpaca itself.
And my second question is, can you elaborate more on how you create the linker? The linker to link the VHH. The linkers. Sorties.
So we use we also there are some sorties that has been published up there, but there are some modifications that we could do to improve the efficacy and the flexibility of the sorties itself. So we created a lot of those peptides and stuff ourselves, like synthetically.
Hello. Great talk, fantastic work. It's really interesting. I just had a question, and this is just me being curious. If there's any change to tumor surveillance when you suppress these cells, or if there's a leftover immune response that can, if there's any change in tumor incidence in these mice, in these models. Tumor? Yeah, if there's a change in tumor surveillance when you suppress these cells.
So
Or if I don't know if people have looked into that at all.
yeah, I guess we haven't really looked into that, but thank you for your question.
Could you use the
But
microphone?
we do have.
I'll use the mic for the recording.
Yes. Yeah, so, sorry. This is kind of a bit awkward. But anyway, I'm a bit too short. Yeah, so for the mechanism of actions that we have seen, we do notice some inhibitory receptor being upregulated, which is usually, you can see like that might be related to like the tumor mechanism aspect you are asking. But what really interesting is that there are multiple mechanism actually at play in this strategy. So that's something that we are still investigating. But we do notice, for example, LAG3 is being upregulated in some of the antigen-specific T cells that we treated in the mice that we treated with this approach. Yeah.
Yeah, thank you.
No worries.
Great, thank you. So I think we should move on to our next talk.
Yeah.
Thank you, Nabrit. Great. Okay. Let's do this. Okay. Oh. It doesn't like my double click. So our next speaker is Inez Patop from Brandeis University, and she is going to tell us about the splicing factor MBL, which forms a regulatory circuit by modulating back splicing in a cell type and isoform-specific mechanism. Thanks.
Okay, hi everyone. I'm Inez. I am a PhD student at Brandeis University, and today I will be talking to you about one of the main projects that I studied over my PhD, in which basically we show that, as my title say, MBL and circMBL form isoform and cell type-specific self-regulatory loop. So I want to start by talking a little bit about circular RNAs. So I think we all here know that as RNA gets transcribed, co-transcription rate gets spliced, basically by a process in which a 5' splice donor will attack a downstream splice acceptor. And we all know this can happen in a linear fashion. It can skip certain exons, and we basically end up having different linear splicing isoforms that we can recognize by the specific splice junctions. And because these transcripts are polyannylated, we can find these splice junctions both in total RNA and in poly-selected libraries. However, a splice donor can also attack an upstream splice acceptor. And this basically generates a covalently closed RNA molecule that we can specifically recognize by looking at this specific splice junction that is not shared by any linear isoform of the same transcript. Particularly because these molecules are not polyannylated, usually we don't see these splice junctions in poly-selected libraries, and we are forced, or we only usually use total RNA libraries. But why do we care about circular RNAs? So basically, we know they are highly enriched in neuronal tissues, in all the organisms that have been studied so far. We know they also accumulate with age in neuronal tissues, also in all the animal models that we have studied so far. And over the last years, our lab and others have found different molecular functions for circular RNAs. But in my talk today, I will only be focusing on the functions they might have by the fact of being just generated. So my work focused on the splicing factor MBL, or muscle blind, that in humans have been associated with myotonic dystrophy, for example. And what our lab showed back in 2014 in cell lines is that the splicing factor regulates its own splicing, generating the most abundant circular RNA in the whole fly head and brain. And because this is so abundant, if the circular RNA comes at the expense of the linear, because usually, or 99% of the time, you either make a circle or a linear isoform, you cannot make both. This can be a way of self-regulating. And basically, generating a function in cis. Something I won't be focusing on this talk today is also the functions it can exert when it gets transported to the cytoplasm that we call basically function in trans. So the main question I try to answer during my PhD is what's actually happening in vivo. And as I told you, this is highly expressed in the fly head and brain, so we basically went back and focused on, okay, what is happening in the fly brain and head just in wild-type conditions. So when we look at the transcript, what we see is that looking back at ribosome footprinting data and also an RNA sequencing data, we see that the most, at the linear level, the most abundant isoforms come for the farthest away and longest UTR, giving rise to two different transcripts and two different proteins that differ by the inclusion or exclusion of this particular exon there, that when it is excluded, generates an upstream stop codon that gives rise to a significantly shorter protein. We call them MBLC and MBLOP because of the name the transcripts have. But what about the circular RNA? So far I've told you about exon 2 basically being circularized. When this splice, donor attacks this upstream acceptor, but this acceptor can also interact with more downstream donors, generating not one, but 4 different circular RNA isoforms. So now that we know what we have, the question is where, because brain and head is a really complex, have really complex and diverse tissues. So we started by looking, so for looking at this, we use total RNA sorted cell types. And basically, by looking at the specific splice junctions, we are really surprised to see that the linear isoforms are actually mutually exclusive. So what we have is that in the photoreceptors, we have MBLOP, while in many different cell types in the brain, particularly super highly enriched in the lamina wide field 2 neurons, we have MBLC. But what about circular RNA? So we did the same, and this was also really shocking. So we saw that, if you remember, I told you the exon 2, so the short circular RNA, it's highly abundant when we look at bulk, and we see that it is expressed in many different tissues, particularly, again, highly enriched in LOF2 neurons. And the other circular RNAs that come from this locus that we believed in the past that were really like lowly expressed, they're actually highly expressed, but super cell type specific. And again, surprisingly, enriched in photoreceptors. So basically, all of these say, okay, we have here about 50 cell types more or less. Can we look at the rest of the brain using, for example, the advantage of single cell data? And the answer should be no, right? Because if you remember at the beginning, I told you that circular RNAs are not polyadenylated, and single cell data is polyadenylated. So we were actually really lucky because circular MBL has poly-A tracks, which allowed it to be present in poly-A selected libraries. And we actually went and looked if we could find the specific splice junction, again, like inverted. And the next question is, how can we envision a bioinformatic approach to actually get this information out? So what we did is we created, or I created basically fake chromosomes. So basically I modified the genome, the genome version that we usually use to align data to be able to capture these particular signals. And we went back to the available single cell data in the fly brain. And if you are not familiar with this type of plots, here each dot is one cell. The fact that 2 dots are together means 2 cells share some type of, in some sense, the transcriptomes, and then we can assign different cell types. Here you can see, for example, the LOF2 cells I was telling you about. And basically, using this data, we were able to see both circular MBL and also the UTR that we were interested in. And of course, first things first, validate that our approach is valid. And we see again that LOF2 neurons are super enriched for the circular RNA, and same as photoreceptors. But we also get interesting new insights like peptidergic or clock neurons. So going back, so I've shown you that in photoreceptors we have MBLOP being co-expressed with different circular isoforms, while in the rest of the brain and also in muscles, we have MBLC being also only co-expressed with circular MBL1. And I told you at the beginning that we show in S2 cells that there is some type of regulation, but the fact that we see them happening together doesn't mean this is regulatory, right? So to prove this, we knock down in vivo each of these proteins, and we assess the levels of the circular RNA. And as expected, we see that when we knock down MBLC, we see a reduction of circular MBL1. If we look this only in heads, only in brain, sorry, we see even a bigger knockdown. While when we knock down OP, we see a reduction in circular MBL2, 3, and 4. So again, this is pointing towards the direction of a self-regulatory loop, but it's not proving it. So far, I've only shown you this part. We know that the protein actually forms or helps the generation of a circular RNA from its own locus. But I have to show you that these circular RNAs come actually at the expense of the linear RNA. If not, there wouldn't be a self-regulatory loop. So to do so, we looked at we use overexpression constructs that allow us to see two things. Number one, that when we have more splicing factor, we have more circular, and that these circular RNA come actually at the expense of the endogenous linear. So we did this, we overexpressed in photoreceptors, and again, two things. Number one, circular RNase goes up. Number two, endogenous linear go down. The same for the brain with MBLC. Circular MBL goes up, endogenous go down. So this led us to our current model, in which we have something happening in the brain and something happening in the eyes. Just as a recap, in the brain we have MBLC that come back into the nucleus and promote circularization of its own locus, generating only circular MBL1. In the eye, on the other hand, we have MBLOP that go back, shuttles back into the nucleus, and promotes not only formation of circular MBL1, but 1 through 4. Something I didn't have time to show you, we also shown that MBLOP inhibits its own splicing. Maybe or not, we are now figuring out if this is actually collaborating with the circularization or not. And to wrap up, I told you about all the functions in cis. In the paper, we also did a lot of studying the functions in trans by using specific shRNAs to knock down the circular RNA in the cytoplasm after it's being produced. And we see a lot of phenotypes, of different phenotypes. I'm only showing you here level locomotion defects, developmental lethality, weeks of phenotypes, and abnormal activity. We did a lot of bioinformatics analysis to try to figure out which cells are being affected by this knockdown, and so on and so forth. And with this, and with just right on time, I want to thank my PI and also the other two people that were highly involved in this project. I am the informatician and also like in the person and so on and so forth, but the RNA work has been done by a postdoc in the lab, NAG, and most of the behavior work was done by Shreya. So happy to take any questions.
Great, thank you. Do we have some questions? Good.
First of all, thank you very much for that talk. It was very informative and clear. I just had a brief question. In the first two talks that we heard, we had a very specific target relating to human health that we were trying to resolve. I was wondering, what are the potential implications for human health of this particular project?
Thank you very much for that question. Something I really want to, I'm not going to be able to because I'm finishing my PhD, but I think it's something we should take forward. So muscle blind in humans has been implicated in myotonic dystrophy. Myotonic dystrophy is a disease that affects muscles, but in the first onset of this disease is actually cataracts. So you have a patient that goes to the doctor at the age of 32, 35, and say, I have cataracts, I cannot see. So I think that all the things that we are showing of the difference between the photoreceptors and the brain and also the muscles, I think it's kind of worthy to keep on studying. I have to say that humans have 3 homologs, MBNLN1, 2, and 3, so it's like 3 genes. MENM3 is mostly fetal, it's in the fetus and in the placenta. The other two are in muscle as and also in immune cells. And all the data that I've analyzed in humans, I haven't seen such a difference like we see here, that we have two isoforms being like, okay, one in the eye, one in the brain. I don't see exactly that, so there might be some overlapping functions, but I think that it will be interesting to move forward. Also studying the circular RNAs in the context of, I don't know, I would like to have some, retina from DM patients or from myotonic dystrophy and non, and see what happens with the circular RNA, for example. I think there are a lot of avenues to go.
It's a very exciting talk, thank you. Is there any evidence that circular RNAs in general, or yours in particular, are translated?
Yes, there is. So we have a paper, our lab have a paper, not me, like the lab have a paper in 2000, I don't remember if it's 16, 17, on translation of circular MBL, produce a really small peptide. The thing is that circular RNAs are already circularized. It's not, like mRNA gets circularized when it's being translated and so on. This is already a circular, so it can only be translated through iris. So it's usually translated under certain stresses because it's not a cap-dependent translation. It's cap-independent. So there are some evidence, like we have that paper, and there is an independent group in Italy that show like this actually in mice for another circular RNA. But there is a big debate in the field because most of them are usually not translated. And the thing is, okay, I think you need to find the right stress for a circular RNA to be translated. Many have potential to be translated, but the fact that they are translated, it's kind of a study in the field.
In your specific case, that small peptide that's made, is that in any way functional or competitive or blah, blah, blah?
Yeah, so back in that paper, so I think they show it, and I don't remember which stress we used to look at it, but there is our lab is studying that, like what happens when, for example, if you mutate the specific something to make it not produce the protein, to see if that changes or not, because actually, and I think this is really interesting, the circular exon 2 is in much higher levels than linear exon 2 in this, in specific cell types. So I think it will be interesting also by itself.
Terrific. Thank you, Inez.
Okay.
And we should move on to our final speaker in this session. So let's So our final speaker in this session is Ernest Taylor from the University of North Carolina, who will talk to us about age and oxidative stress regulate NRF2 homeostasis in human articular chondrocytes. Thanks, Ernest.
Thank you. Yeah, so today I'm going to be talking about how oxidative stress regulates NRF2 and how that affects human chondrocytes, and potentially how that affects osteoarthritis. So I'm doing my postdoc in UNC under Dr. Richard Loescher. So aging in OA, so what we know is around 32 million adults suffer from OA, and 15 of those individuals reported severe pain, joint pain due to arthritis. And what we also know is that arthritis is one of the leading causes of work disability in the United States. So here, we actually get a lot of our samples from the Gift of Hope from donors. So what we did here, we got some young, normal donors, what we classify normal, no degradation, normal cartilage. And what we did here is some histology. As you can see in these sections is some safran O staining, which is going to stain the proteoglycans. As you can see within the normal tissues, there's a high thickness of cartilage that's there. So when the joints are rubbed together, femur and tibia are we're putting mechanical load on those two joints, you're getting that cushion in between those two joints. But as you get older, you get, due to mechanical loading, as you get older, you see thinning of that joint, but there's no loss in the integrity of that joint. But once you get OA, you lose that superficial zone of that lining of the cartilage, and also you get less cell number in the articular surface, and also you get problems with the underlying bone. So some of the challenges that arise from trying to treat OA is that OA is a disease of the whole joint. It just doesn't affect the articular cartilage, it also affects the synovium and also the synovium fluid. And another challenging thing is you really don't see patients until they are experiencing that severe joint pain, and it's usually around this stage here. So it's hard to kind of reverse that effect that you're seeing in the articular cartilage because one, for the most part, the articular cartilage is very avascular joint, so it doesn't have relatively good blood supply, so it's hard to regenerate those chondrocytes within that cartilage. And one of the there are a lot of causes to OA, but one of the main causes to OA is ACL injury, and that's leading to joint reconstruction. And this is kind of a problem that we're seeing in young athletes, because the young athletes now are getting ACL injuries at younger ages, and when that happens, most of them will get joint reconstructions. And what a lot of studies have seen, 85% of those individuals do, within a specific time frame, develop osteoarthritis. And currently, there are no disease-modifying drugs that exist. So what we try to do in our lab is try to find targetable proteins and look at those signaling pathways and see if that would be a targetable drug that we can potentially target in this disease. So in this talk, I'll be focusing on oxidative stress. So aging and extracellular signaling, such as catabolic growth factors, matrix fragments like fibronectin fragments. Fibronectin fragment has been shown to increase MP13 and other cytokines like IL-6 and also mechanical stress and other cytokines like TNF-alpha. And so what that happens is that it increases hydrogen peroxide and other reactive oxidation species within the mitochondria and also within the cytoplasma. And what happens is you get decreases in antioxidants. And particularly one of the main antioxidants that I focus on within this project is NRF2. And NRF2 is an important antioxidant because not only is it an antioxidant, it also, when translocated to the nucleus, it helps in the transcription of other antioxidants. And we know that oxidative stress disrupts signaling of MAP kinases, IGF, and the AKT pathway, which is important in cell and cartilage proliferation and maintaining articular cartilage cells. So when you get that disruption from oxidative stress, you get aging phenotype, chondrocyte death, and also cartilage matrix loss. So under physiological levels, ROS is good because you maintain that cellular homeostasis, but at higher levels, you get oxidative stress. So the purpose of this project was to investigate the oxidative stress regulation of NRF2 in human chondrocytes. And we get our young and old donors from the Gift of Hope. We get the femurs. And from OA donors, we get those after knee replacement from UNC ortho department. And secondly, we wanted to examine the effects of NRF2 depletion and NRF2 activation on these human chondrocytes. So as you can see, the protein levels in older and OA individuals were lower than that in younger individuals. As you can see, there was a significant decrease in NERV2 expression in the older NOA. So what we wanted to do next is look at under oxidative stress, how does NERV2 respond in young and older individuals? So we accomplished this by using metadione, which increases the hydrogen peroxide levels in both the mitochondria and cytoplasma. So you can see in the young individuals, there is a significant increase in the NERV2 expression, while in older individuals, we don't see that increase in a significant increase in NERV2 expression when we subject these cells to oxidative stress. So what we're doing down here is that for NERV2 to become activated and increase the expression of other antioxidants, it must be translocated to the nucleus. So what we did here, again, we were treated with metadione for 15 and 30 minutes. And as you can see in the older individuals, when there's oxidative stress within the chondrocytes, you see an increase in NERV2 expression in both the cytosolic and nuclear fractions, and we don't see that increase, that significant increase in the our older donors, which have lower levels of NERV2 expression. So what we wanted to do next is look at a NERV2 activator, RTA408, and see how it affected other NERV2 antioxidants that are targeted by NERV2. So as you can see here, when we treated with RTA408, the NERV2 activator, at 2 different concentrations, we saw a significant increase in NERV2. SRX, which is another target of NERV2, NQ01. We saw increases in both of those, but we didn't see an increase in PRX1 or the thioredoxin interacting protein. Thioredoxin interacting protein, it kind of works in reverse. What it does is it increases ROS production and it induces oxidative stress. And we also looked at P65 NF-kappa B, and we didn't see any changes in the phosphorylation or the total protein levels of P65. And here, we repeated this in the OA chondrocytes that we get. As you can see, it followed the same trend, increasing NERV2, SRX, and NQ01. But interestingly enough, we saw decreases in the thioredoxin interacting protein. And also what we saw different with these OA chondrocytes is we saw decreases in P65, but that was mainly due because we saw increases in the total levels of P65. And we were expecting to see the opposite of that. But what we also did see is that the total levels of P65 were increased in our younger and older compared to our OA. So kind of to address this issue, what we're doing now is looking at nuclear and cytosolic fractions, because P65 becomes active once it's in the nucleus. So we're going to look at those nuclear fractions and compare those, the OA samples to both the young and the older. Here's just to kind of confirm NERV2 knockdown in this, how it interacts with those SRX and heme-oxygenase and NQ01, just to see if that NERV2 activator was specific that we were using, or did it have off-target effects. So as you can see, NERV2, when we used the siRNA to knock down NERV2, we saw decreased levels of NERV2, decreased levels of SRX, heme-oxygenase, and also NQ01. We saw decreased levels in all of those. And we didn't see any changes in PRX or the thioredoxin interacting protein, which is kind of different from what we saw in our OA cells. Not sure exactly why that decrease was seen in OA cells, but not in normal chondrocytes. So we're kind of just looking into that to see why that may be happening. And lastly, what we wanted to do here was look at how NERV2 affects certain genes that are important in articular chondrocytes. So we looked at agarkin. Agarkin is important in regulating the production of proteoglycans. Proteoglycans basically are important in osmotic properties, and it gives that structural integrity to the articular cartilage. And also we looked at type 2 collagen called 2A1, which again adds that structural matrix to the cartilage, and it gives you again that cushion and strong support for your articular cartilage. So as you can see here, when we just treated with the RTA compound alone, we didn't see any increases in aggregate or called 2A1 in either our OA or our normal. IGF is known to increase both aggregate and called 2A1, but what we know is, and what you can see here is IGF-1 didn't have any effect on aggregate and called 2A1 because in osteoarthritis, that pathway is reduced because there's reduced signaling of AKT. But when we treat it in combination with the RTA and IGF, you can see increases in both aggregate and called 2A1. And this could be due to the fact that the NRF2 activator is inhibiting the ROS production that is affecting AKT signaling within the IGF-1 pathway. So this study implicates age and OA as a determinant of NRF2 homeostasis and chondrocytes. The RTA 408 NRF2 activating compound increases NRF2 expression, and in turn increases expression of other antioxidant genes. And we know that activating NRF2 protects against ROS-induced resistance to IGF-1 signaling. And targeted therapies aimed at maintaining NRF2 activity could be useful in maintaining chondrocyte homeostasis and maintenance of intracellular antioxidant functions. Here's my lab. I just want to acknowledge my lab, our funding sources, and our tissue donors, the Gift of Hope, and the UNC Ortho Department. And I'll take any questions.
Hello. So this drug that you are trying to use as a prevention for the disease, or it would be used when the patients already get some symptoms and yeah, that's it.
So the RTA compound alone would not be sufficient at treating OA. It could possibly make the environment within that joint better, and we can add something that increases chondrocyte production in combination with that. But the RTA compound alone wouldn't be sufficient enough in treating OA, but it could prevent the increased oxidation within that joint.
So NRF2 is also a target in cancer, and there's like downregulation models that prevent like kidney cancer and things like that. And so I'm just wondering, like with the NRF2 activator, is that potentially a problem? And how would you deliver it? Can you deliver it locally? And if you delivered it locally, would that circumvent any issues with potential cancer?
Yes, so that's a good question. So since the joint itself is very avascular, and I've done some pharmacokinetics on other drugs that you usually enter into the knee joint don't usually enter systemic circulation. So it could possibly be, it probably wouldn't at those small levels, if it did enter systemic circulation, affect other tissues within the body. Good morning.
Your talk stimulated lots of thoughts in my head, so I'm sorry, my question is tangential. Hypoxia-induced factor, HIF, is expressed in low oxygen conditions. And when you started talking, you mentioned the avascular environment, which is a problem in terms of regeneration and so forth. So does HIF get activated in joints, especially during this OA process?
Yes, it does get activated within the cartilage. It does, yes.
And so that turns on a lot of the HIF downstream factors. Does that influence in any way recovery?
I I don't know. It could though. I'm not sure. That would be something I kind of look into because we haven't did anything many experiments in terms of NER2 and HIF. So I wouldn't know.
I have other thoughts, but I'll catch up with you later.
Okay, thanks.
Thank you.
Great. Thank you, Ernest. So please join me in thanking all of the morning's speakers for a really terrific session. And I think we're going to take a 5-minute break. Does that sound good? To get everything set up for the panel. So see you in 5.